Welcome everyone to the recorded webinar series on LED-based photoacoustic imaging. My name is Mithun and I'm a Research and Business Development Manager at Cyberdyne. In this series, several key opinion leaders in the field of photoacoustics will talk about LED-based photoacoustic imaging and its preclinical and clinical applications. Today's interesting talk will be about imaging aspects and applications of LED-based photoacoustic and ultrasound tomography and will be delivered by Professor Wiendelt Steenbergen from University of Twente in the Netherlands. Professor Steenbergen obtained his PhD degree in fluid dynamics from Eindhoven University of Technology in the Netherlands in 1995. In 2010, he became full professor and group leader of the Biomedical Photonic Imaging Group of the University of Twente. Since 2017, he is Vice Dean Research of the Faculty of Science and Technology. His current research interests are speckle-based perfusion imaging, photoacoustic imaging for rheumatology and chronic wounds, quantification of photoacoustic imaging using acousto-optics, and thermal imaging of tissue perfusion. In this exciting webinar, he will talk in detail about tomographic imaging using an ultrasound and LED-based photoacoustic system. With that, I will turn it over to Professor Steenbergen.
Hello, I am Wiendelt Steenbergen of the University of Twente in the Netherlands, and I want to share with you some of our results obtained with an LED-based photoacoustic and ultrasound system. First of all, we acknowledge the National Center for the Replacement, Refinement and Reduction of Animals in Research for funding parts of this work, and I thank my colleague Dr. Francis Carlo-Joseph for obtaining the results presented here. What can you expect from this presentation? First of all, I will address the issue of why tomographic imaging. And next, I will go into some aspects of the system design. And of course, I will show you some of our results of imaging finger joints and small animals. Let's start by looking at one of the advantages of photoacoustic imaging. The ultrasound transducers used in photoacoustic imaging can also be used for ultrasound imaging. From a clinical perspective, this means that photoacoustic imaging can be typically used in applications where ultrasound imaging is already in use. An interesting aspect is that by simply plugging in a pulse light source, conventional ultrasound imaging systems can be converted into photoacoustic systems. One disadvantage of using conventional ultrasound systems with a linear array for photoacoustic imaging is the directivity of the transducer, resulting in a limited view. This is clear in the photoacoustic image of a leaf skeleton, as shown here, where a linear transducer was used to image from two different angles. And for each different angle, we only see a part of the phantom. However, many applications, including finger joint imaging, small animal imaging, breast imaging, etc., allow for a larger view angle. In these applications, tomographic imaging can provide more information than imaging using a linear array. For photoacoustic imaging to explore these clinical and preclinical applications, there is a need for cost-effective and compact tomographic systems. Additionally, systems with minimal operator-dependent and eye-safe systems, such as using an LED, are definitely an advantage for fast translation. Linear transducers were one of the first choices for tomographic imaging in photoacoustics. Already a long time ago, Orjevsky demonstrated tomographic photoacoustic imaging with a linear array scanning around a sample. This was further explored by Kruger by combining both ultrasound and photoacoustic tomographic imaging. Other groups, including Leon Wangs and Tia Christos, also use tomographic imaging using a linear array. Linear transducers have a higher yield and are low cost compared to custom developed transducers. In addition, they are easy to integrate with the system for combined ultrasound and photoacoustic imaging. Another factor than the ultrasound transducer that makes photoacoustic systems costly is the pulsed laser source. Most of the tomographic systems use expensive and bulky nanosecond pulsed lasers. More affordable and compact light sources, such as laser diodes and LEDs, have made significant progress in photoacoustic imaging in recent years. Compared to laser diodes, LEDs give a wider choice of wavelengths in the visible range, and they can make a compact system. Allen and Beard have demonstrated using LEDs for tomographic imaging using a single element transducer. Recently, Tanya's and Kotapalli's groups also showed the feasibility of using LEDs for photoacoustics. Here we present photoacoustic and ultrasound tomographic imaging using a linear array and LEDs for illumination. The system that we used is the Acoustic X by Cyberdyne, and we will go into various imaging aspects and show some applications. The linear ultrasound transducer that we used has a frequency range of 4 to 10 MHz. The pitch between the elements is 300 micrometers, and the array is covered by an ultrasound lens with a focal distance of 20 millimeters. The opening angle per element for sending and detecting ultrasound is almost 27 degrees. For tomographic imaging, we can have two transducer configurations. If we scan with the short axis of the transducer going around the sample as shown here, we can ideally form a 3D image. And if we scan the sample using the long axis, we will get a 2D image as shown here. However, the focus of the transducer can degrade the image quality in the 3D case and requires a combination of linear and circular scan for 3D imaging. In this work we have addressed only the two-dimensional imaging, with the possibility to extend this to 3 dimensions by stacking multiple slices. In tomography, an important question is how many angular views are optimal for tomographic imaging, also taking into account the imaging time. To answer this question, we did a simulation study. We developed a numerical phantom with structures with different thicknesses, orientations, and absorption levels. We simulated forward acoustic wave propagation using the K-wave toolbox and reconstructed the images using a Fourier domain algorithm. This is the reconstruction on the basis of one single view, where we observe that the number of lines is not well visualized. Increasing the number of views until 64, we see a stepwise image improvement. We then compared the reconstructed image quality using the so-called Structural Similarity Index, SSIM, and the peak signal-to-noise ratio, PSNR. It was found from the simulations that 16 angular views are optimal, which is 22.5 degrees step. For the experiments, we decided to use 18 views, which is a 20-degree step size. This is a slight oversampling compared to the calculated value. Next, something about the configuration of the LEDs used for illumination. Here we used 4 LED arrays, each array consisting of 36 times 4 elements, and arranged in 2 different configurations. In the first configuration, the LEDs are fixed on top of the sample, as shown in the figure. The transducer is then scanned around the sample for tomographic imaging. This configuration can be used for ex vivo samples or brain imaging, where illumination from the top of the sample is possible. In the second configuration, 2 LED arrays are placed on either side of the transducer along its long axis and directed towards the focus of the transducer. Additionally, 2 more LED arrays were placed in the imaging plane with a 105 degrees with the transducer and a tilt of 5 degrees with the imaging plane. This configuration was designed to image objects with a maximum diameter of 4 centimeters, and also to minimize reflected acoustic signals reaching the transducer surface. This configuration can be used for applications such as small animal imaging or finger joint imaging. To test these configurations of the LED arrays, we performed a numerical study, and we did both optical and acoustic simulations. The light propagation was simulated using Monte Carlo, using the LED specifications and a phantom mimicking soft tissue. And here you can read all the specs. The acoustic forward simulation was performed using the K-Wave toolbox, and reconstruction was done with a Fourier domain reconstruction algorithm. Acoustic properties, including the speed of sound, the density and attenuation, were modeled. And in addition, 2 different speeds of sound were used for the phantom and the coupling medium to see its effect in the reconstructed image. In reconstruction, a constant speed of sound was used. The tomographic image was formed by spatial compounding of B-scan images obtained from 16 different angles. These are the fluence maps from top and side illumination. The 1/E depth gives an indication about the achievable penetration depth. A vascular phantom was developed from the digital retinal images for vessel extraction database, the DRIVE database. These are the initial pressures for a single illumination position obtained by multiplying normalized fluences and ground truth for both the top and the side illumination. We see a clear difference between the top illumination and the side illumination, with mainly the region of high fluence rate emphasized. Based on 16 angular views, this tomographic image is obtained from top illumination and side illumination. The reconstructed pressure is higher at the center for the top illumination and higher at the rim for the side illumination. The change in speed of sound caused some smearing of edges. This is further evident in the line profiles. Spatial compounding of B-scans is an easy way for tomographic imaging, but selection of the speed of sound is important in this aspect. Now we have explored the effect of the amount of views and the various illumination patterns. Let us go to some experimental results that we obtained. Let us check the leaf skeleton again. B-scan images were obtained from multiple angles and are then spatially compounded to form the tomographic image. Here we clearly see the development of the image when more and more views are being added. In the leaf skeleton we have 4 levels of structures. This is visible in the zoomed in image of the leaf. Only the smaller structures in the leaf were undetected due to the band-limited transducer. But all the other structures with 3 different thickness levels are well reconstructed. Using a linear transducer in a single view, we obtained a lateral resolution of 0.47 mm and an axial resolution of 0.22 mm. The smaller structures in the reconstructed leaf image had an average thickness of 0.26 mm. This clearly shows that there is an improvement in resolution along the lateral direction, and this comes with a trade-off in the axial direction. Now we go to the imaging of joints in small animals. This is an example of tomographic imaging using top illumination. Here we imaged a mouse knee. First, have a look at the ultrasound images here. Figure B was obtained using plane wave ultrasound, and figure C here was taken by conventional line-by-line ultrasound imaging. In both cases, a tomographic image was obtained using spatial compounding of B-scan images from 18 angular views. The B-mode ultrasound can achieve a better image quality in terms of the contrast between the tissue and the bone, but this comes with a slightly longer imaging time. The photoacoustic image shows the blood vessels around the knee, with a large blood vessel visible in the photograph, partially in the imaging plane. Using the LED-based ultrasound and photoacoustic imager, we created this finger joint imaging setup with the LED bars in the side illumination mode. For tomography, the finger is placed in the construct of the ultrasound probe plus the LED bars, who then rotate around the finger. Everything is submerged in water. We imaged a finger joint in a healthy volunteer. Figure A is an overlay of maximum intensity projection images of a stack of ultrasound and photoacoustic tomographic images. The other figures, this one and these ones, are tomographic slices 5 mm apart. The photoacoustic images show skin and blood vessels. There are reflection artifacts like here and here from the bone in the center of the photoacoustic images. In some cases, blood vessels in the photoacoustic images do coincide with hypoechogenic regions in the ultrasound images, like here and here. Next, we go to small animal imaging. In this stage, we can only demonstrate images of post-mortem samples. We used side illumination and placed the animal in a special 3D printed holder. The animal was a euthanized male Belp sea mouse. Full view tomography was done for obtaining photoacoustic images of livers after induced liver fibrosis. Here we compare various single views with full view tomography. In each of these 3 single view images, other objects can be observed with different resolutions depending on the viewing direction. In 16 view tomography, we can see many organs at the same time, with an imaging depth of about 10 mm. A feature at a larger depth, like this here, may be a reflection artifact due to reflection of photoacoustic sound on the bone structure of the spinal cord. Images like these show that it is essential to have both the photoacoustic and the ultrasound image. While the photoacoustic image alone is difficult to interpret, the ultrasound image casts a much better light on the anatomy of the object. So you could say the photoacoustic image gives the function associated with the presence of blood vessels, and the ultrasound image gives the structure. The animals in our study were divided in a group with carbon tetachloride induced liver fibrosis and a control group. To reduce the imaging time, tomography was limited to 3 angular views, and of each animal 10 slices were obtained in a region spanning 50 millimeters, visualizing the liver region. Now take your time to compare the images of the control group and the disease group. And the overall observation is that the photoacoustic response in the fibrotic livers is larger than the photoacoustic response in the control livers. Here we give a more quantitative analysis of the results. Based on the ultrasound images, we have delineated the liver region. And this again illustrates the importance of having an accurate co-registration of photoacoustic and ultrasound images. In the ultrasound images, we see a so-called hepatic nodularity, and both the mean echogenicity and its variance are significantly larger in the diseased livers than in the controls. In the photoacoustic images, we clearly see the enhanced light absorption caused by neovasculature. Also for photoacoustic images, the contrast in the photoacoustic images is larger than in the control images. While ultrasound images alone already show a distinction between fibrotic livers and controls, the photoacoustic images will give unique additional information related to the growth of new blood vessels as a part of the pathology. The histopathology, in which a collagen 1 immunostaining was used, clearly shows the increased presence of collagen bridges in the fibrotic livers. It is time to come to a conclusion. We demonstrated combined ultrasound and photoacoustic imaging using a standard linear ultrasound transducer and LEDs for illumination, and we have observed an imaging depth down to 10 millimeters. The ultrasound transducer and the LEDs can be combined in a compact and affordable ultrasound and photoacoustic scanner, such as shown here with the Acoustic X. Various configurations for sample illumination have been explored, and we established the number of angular views needed for good tomography using the linear array. Furthermore, we focused on potential applications in finger joint imaging, which may be relevant for rheumatoid arthritis imaging, and liver fibrosis as an example of small animal imaging. Future work will be dedicated to improving the imaging speed. It would be great if Doppler and power Doppler options in the ultrasound imaging will be added, and we will explore the imaging of alive small animals. For further reading on our work on ultrasound and LED-driven photoacoustic imaging, we refer to our recent papers in Biomedical Optics Express and Sensors.